And so in learning about cell division, we have to go through these three segments: interphase, the second one being mitosis, and the third one being cytokinesis. And so G1 phase, S phase, and G2 phase, all components of interphase. I always remember this acronym, IPMAT. And so interphase, and then the cell division are going to be the PMAT. And so you can see that PMAT represents prophase, metaphase, anaphase, telophase. And then that last bit is cytokinesis, which isn't too tough to remember. And so G1 can be considered sort of a growth phase. You can consider this G as growth 1. You can consider this G2 as the second growth phase. And S phase is going to be the synthesis phase. And so originally we were supposed to go over DNA replication in this segment, but I'm going to add it to our embryology section after. And so we just have to keep in mind that DNA was a molecule inside of the nucleus that if it wants to create one cell to create a brand new cell, it needs to create all those instructions. It needs to replicate it for the other copy of that cell. Make sure to grab grab a packet. And to grab, it has to make the same genetic information from one cell to the next cell. And so to do so, we have to think about what has to occur during these phases, and that G1 is going to be responsible for duplicating its organelles. Remember all those organelles we learned about? We learned about the nucleus, the endoplasmic reticulum, the rough and smooth. We learned about the mitochondria, lysosomes, Golgi body or Golgi apparatus, and all of those have to be made for the new cell that's going to be created. And so we have to duplicate those organelles. Make sure to know for our exam which organelles do what, and make sure to know whether they're membranous or non-membranous. So with that, S phase. Now, S phase is going to be the synthesis phase where the genetic content is going to be replicated, the DNA. And so we don't need this fact right now. We'll add this when we get into embryology. But otherwise, we already know that DNA serves as the instruction manual for the cells. So it has to make logical sense to us that if we're going to make a brand new cell, they're going to need a copy of that instruction manual. And that's what S phase is to replicate the DNA. That's all we need to know regarding synthesis phase. The G2 phase can be considered as a second growth phase, and this is where enzymes and increased protein synthesis and the high energy demands are going to be met for the process of cell division, which is mitosis. And so we have to make a lot of ATP, and what organelle is going to be responsible for synthesizing a lot of our ATP? Mitochondria. And so now here mitosis is going to be prophase, metaphase, anaphase, telophase. And as we progress into those, just keep in mind that the act of dividing, going from one cell to two, is only occurring under mitosis. And then everything is written here for you, exactly what was said. But just keep in mind, last thing to add here, is that when we have one cell dividing into two cells, we consider the cells that originate from that original cell, we consider them daughter cells. And this image doesn't summarize kind of a complete picture that I kind of want to mention too, is typically whenever you have this, especially in the context of skin, is that you always have one cell making a clone of itself, but one is going to stay back and the other one is going to go serve as the lineage of cells. And this one's always going to stay so that way it can always undergo mitosis and create a new cell as a result. But that's never really depicted in these diagrams. But for simplicity's sake, I understand. One original cell, it breaks apart into two daughter cells. And cytokinesis is finally the act of them dividing it up. So with pictures, it's a little bit easier. And so when we look at this diagram, try to keep in mind that we are going in order here. We have prophase, metaphase, anaphase, and telophase in that order. And so we have to sort of understand what's going on from going from one cell to going to multiple or two daughter cells. So first phase, prophase. Let the P remind you, I kind of think about it as a prepare phase more than I do actually before. But preparatory phase, where the nuclear envelope disintegrates. And this has to make sense, right? Our nucleus has to release that genetic information so that eventually the other cell can also have a copy of it. But the nucleus is bound or keeping it bound together. So first thing we got to do in prophase is we got to break apart that nuclear envelope or the membrane of the nucleus. the chromosomes. And although we don't necessarily know what chromosomes are just yet, what our DNA is situated as is in this picture here. Your genetic information, your DNA molecule is this helical structure that is always protected. I'm going to do it in a different color, and I'm going to highlight it like this. There's going to be a protein enveloping it and covering it up like a glove. And whenever your body wants to read, say, genes 1 over here, I'm just going to write a 1 just for the sake of example, and 2 here, that when your body wants to read gene 1 or this segment of the DNA, it'll unwound this protein and it'll expose these little regions and it'll read it as an example. So these chromatids is a complex of DNA and protein, and that's as much information as we need as far as chromosomes and chromatids. DNA plus proteins. And so DNA and proteins are always together. Your DNA molecule isn't just floating around in the nucleus. It has little proteins designated to keep guard and to open it up whenever it needs to be interpreted or read. And so when we say that the chromosomes coil and become visible, all we mean to say is that the DNA complex, as we can see in this nucleus, that the chromosomes start condensing, and then you can see the DNA molecules and their proteins get wider. They get larger. And that's what we mean by condensing. You can see from this picture to the one on the right-hand side, you can see that the chromosomes have condensed, or the DNA molecule with its protein complex is now much more visible. And so replicated, don't worry about centrioles today. Oh, yes, we do. Sorry, confusing this with something else. And so centrioles, now we have a function for our centrioles, and I'm going to jump to it. Might as well review a little bit of our previous material. That make sure you know when we go to the cell, you need to know the anatomy of this, be able to identify these structures off of this picture, and make sure that you know the function of each one of them. Now, one of them that may be a little bit difficult to understand coming from the perspective of an intro anatomy portion, right? Because we don't really know exactly the little nitty-gritties of DNA, right? Or RNA or proteins, and what exactly cell division was at the time. But now we saw that centrosomes were always functionally defined as something that organizes the cell. They're typically talked about as organizing centers. And so this outer region is a centrosome. The little tiny rectangular ones are going to be your centrioles. So I'm going to draw that on your other notes so that way you have a little bit of a reference to add to this. So right here, you can see these are going to be the same objects we just saw. The circle is going to be your centrosome, and the little tiny cylinders within that usually are pretty perpendicular to each other, those are called centrioles. So in red here, this is going to be our centrosome. Let me lay this flat. There we go. Centrosome, and in black we have our centrioles. And as far as we're concerned, they are almost the same thing, in the sense that their role is going to be to move the DNA complexes. And from now on, instead of saying DNA complexes, I'm going to say the word chromatids or chromosomes, okay? So make sure that we know when I say chromosomes for our intro section, you only need to know that that means the DNA protein complex, the instruction manual of the cell, okay? And so with that, our nucleus is breaking apart, and our centrosomes have to start guiding those chromosomes. You'll see that throughout this segment, you're going to see that the rest of the phases can be identified simply by the location or how those centrosomes and centrioles are organizing the genetic content or the chromosomes. And so let's get into it now instead of an overview and then going back to the beginning. So when we say replicated centrioles, now what this means is that you start off with one of these, and then they have to make two of these, because again, we're going to make two different cells, so one of them needs a little guiding light to make sure that these DNA molecules are going to the right side, and then this guide is going to guide the DNA to the other polar end. And so that's what this little statement meant, is that we're going to need to move those chromosomes around, so we're going to need another copy of those centrioles. And we can leave this fact out today and we'll add it to our embryology section. So go ahead and cross this out. We're going to keep it simple, chromosomes. And so with that, now metaphase. Metaphase can always be clearly defined. And I'm going to go ahead and label these because these are sort of oddly placed. This one's prophase. And so is this one. So I'm going to put a bracket. This picture doesn't really define that. But otherwise, that whole segment is prophase, both of those. And how I can tell just at a glance is that M, I usually let it remind me of the word meat. And so you can see that down in the center here of these cells that the chromosomes were pushed down to the center in what we refer to as the metaphase plate, which is sort of this midline down the center of both of these dividing cells. And so that metaphase plate, again, will have the chromosomes in one line. And you can see that here on this image, that's our metaphase plate. If we look at this next segment, and I'm going to go ahead and make sure that this is labeled appropriately, metaphase, you can see that anaphase is going to be when this portion goes apart. You can see that these chromosomes begin to divide apart or to opposing ends, to polar ends. And so I let this A remind me of what it already represents in Latin, apart. So the chromosomes get pulled apart. They are no longer along that metaphase plate. Another statement that we're not going to need, and might probably add this complication into embryology, but you can take this segment off here and just keep it simple. How would you recognize it? You know that the chromatids separate. The chromatids separate. And now telophase. Telophase, I sort of let it remind me, in Latin means end, so you can use that one, or I sort of use it to remind me of like tear apart. How do you spell tear? Thank you. I was going to write tear, but that's T-A-R-E. That's when you weigh something and you displace it from zero. So I wasn't sure. Anywho, so with that, telophase is going to be tear apart. So that means that you have one cell now ready to divide between the other, but it hasn't quite done it yet. Instead, what you get is sort of a pinching off. Another appropriate word is that you see an invagination here, a sort of a groove opening on these ends. That groove is referred to as cleavage furrow. And that's really what defines telophase, is when those cytoplasms begin separating. And so that invagination is a result of that. And the word is cleavage furrow. That's what that space in between is. And then finally, during cytokinesis, the final chapter in this whole process, is when the cell actually begins to separate from the original cell. And now you have your two daughter cells with two distinct cytoplasms and two separated plasma membranes. And so that's our process of cell division that we need to know. So keep it very simple. Keep just quick definitions of what each phase is, and just know the definition or what chromosome means, that it's just a DNA and protein complex. And when we get to the genetic portion of our embryology, we'll talk about it in a little more detail. So with that, I believe that's the end of this. Right. We went over connective tissues, right? We finished this segment. I know that I'm repeating the mitosis stuff, but it gets left out. So that's why yes, I remember your question about glial cells. All right. So with that, the reason I like to actually start this lecture off with mitosis is because the integument system is a mitotically active area. It's the site that we've been seeing are stratified squamous epithelia, which has a high mitotic rate, meaning that the cells down here are always going to be undergoing prophase, metaphase, anaphase, telophase, and always generating new cells to go and serve as a little dead cell on the surface of the skin. So a little bit of this you already know. And with that, When we look at this segment here, you can see that you can divide the entire integumentary system from cutaneous membranes and accessory structures. Cutaneous membrane is going to include the epidermis, which you kind of already know what it is. And let's use this flow chart a little productively. This is going to be epithelial tissue. Specifically, if it's in the outer skin, what's the type of stratified squamous we have? Perfect. Keratinized, stratified, squamous. And an inner layer, which we defined as non-keratinized. Dang it. And so when we talk about the epidermis, we are encountering the outer keratinized stratus. Other than that, we do not care about this one. This one is found in the inner body. And keep in mind that the integument system is regarding your outer skin and all the structures that come out from it, but you have learned that tissue already that makes up the epidermis. That's going to be the keratinized stratified squamous. On your lab exam, you will see the possibility of being as non-keratinized, but that's not going to be the subject for integument, so now you're only going to be seeing this one explored. So this word will make a lot more sense to us moving forward. Dermis is going to have two little underlying layers. One's going to be called the papillary layer, and the other one's going to be called reticular layer. And this is going to be the area where we have connective tissue. Nervous tissue. Oh, dang it, wrong thing. We have nervous. And even muscle, specifically smooth. So you can see how everything you know about connective tissue is going to be seen today, put together like a puzzle. And then we have our accessory structures. And if we think about exocrine, which tissues love to serve as glands? What's the shape of them? There's two that like secretion. Cuboidal and columnar. And in this case today, the one that we're going to encounter the most, and only really, is cuboidal. So you can see even embedded within all of these tissues, you can see another epithelial tissue, cuboidal. And the nails are going to be their own sort of a case study in a way. And let's keep going. All right, just some fun facts. So your 16% of your body weight is approximately coming from just your integument system, your hair, your skin. It's about 2 meters squared per surface, a meter squared per surface area. It is your first line of defense. It is like a suit of armor. It's protecting you from the external environment, and it is considered an organ. And so your skin is your largest organ technically, and your largest organ system, or most visible and accessible one. And you can divide it again between cutaneous membranes and accessory structures, just like we saw in our flowchart in the beginning. And so starting in the beginning, the cutaneous membranes, the more superficial components, we have our epidermis and our dermis. Our epidermis is going to be made of stratified squamous epithelia. Don't forget to add the note keratinized. Keratinized. Our dermis is going to possess connective tissue. We saw that it has two layers, papillary layer, reticular layer, and you can see papillary layer is going to be made of areolar connective tissue, the one that has collagen and a little bit of elastic. It's made by fibrocytes. And then we have reticular layer, which has dense irregular, which is also made by fibrocytes, which is also secretes collagen, but they are two entirely different. One is a very loose connective tissue. The other one is a much more dense tissue, so they have different textures. And if we think about their overall role now, or their placement in our cutaneous membrane, or a little diagram here, then when you look at this piece of cake, as you approach the integument skin, this skin cake that you're seeing here, you want to learn these structure names and the organizations before you get into the nitty-gritty. Save yourself some time and just learn all these terms and be able to ID them first, and then try to figure out what they do in the order of the notes. It'll make it a lot more easier in general, for the remaining course. That's typically the best way to approach things. Memorize the figure on the page, then apply the context that's on next to the picture. So with that, our epidermis is only going to be the superficial layer at the very top, and then it's going to be separated by a lamina propria, or you know it as the basement membrane. The basement membrane. The basement membrane separates the epithelial tissue from the connective tissue. So everything on top of that line, or in purple blue, all the way to the top is nothing but epithelial tissue. The biggest giveaway that the dermis has an insane amount of connective tissue is going to be these blue vessels and these red ones. What do these represent? Capillaries. And capillaries are a form of what type of vessels? Blood vessels. And blood is a type, what family of tissues? Connective tissues. And what type of cells are in blood? White blood cells and red blood cells. And so you can see that the dermal layer is going to be vascular and the epidermis is avascular. So you can see that there's not blood, or there's not capillaries going into that segment. Now we mentioned here the dermis, that the papillary layer is in possession of what connective tissue? Areolar. And the reticular? Irregular. And what I like going back to our connective so we can review, and also we can easily understand this a little bit better, that if we start in order, this was your overall, the outline that I drew for all of you in lab, which represents everything you need to know for connective tissues. And you'll notice areolar tissue was categorized as loose. And dense irregular is the other one in question, categorized as dense. Now, if we analyze the pictures of both of them, this is a nice little drawing of areolar connective tissue. And areolar connective tissue has a lot of ground substance, or a lot of fluid space. And what were all these asterisks representing? I mean, what I told you all not to worry about it too much, but what were they representing? White blood cells. And so this space needs to have an insane amount of fluid so that white blood cells can navigate around and patrol for infections. So doesn't it make sense that the papillary layer is going to have areolar tissue, which is a very loose fluid connective tissue, on this first layer of our little skin cake that we have on the screen, right? This little layer. And it's not called the skin cake. I'm just joking when I say that, by the way. And don't say that outside of this class. But it just looks like a cake to me, so I always just look at it that way. And anyways, and so this layer here is our papillary layer, and made of areolar tissue, which is going to have a lot of white blood cells. So in the case you get a cut, you will not, or you will be able to fight off an infection. And so that's why areolar tissue is the best material for that segment. If we compare it now to the other one, dense regular. We saw in lab that you're only being tested in lab for dense regular, which forms ligaments and tendons, a very tough material that has all its collagen laid out in a very convenient parallel function like this, configuration, pardon. And when you look at irregular, you can even see that it's already denoting where you're going to find it in this reticular layer, because it doesn't have the same arrangement as collagen as this, so we call it irregular. And so this type of network of collagen allows for a little bit more elasticity. I know that I don't like using that word because we have an elastic tissue, an elastic fiber, but that's really what it does. And that's the reason that when I pull my skin here, that it doesn't rip off like a piece of paper. Like this is why it can stretch out, or anywhere you pull, this is why. And it's your irregular that you have to think. It has a more of a rubber band texture to it in the sense. And so that allows for that little bit of stretchiness, and that way your skin can retain some ability to reform its shape after a little bit of maybe some wear and tear, some excessive friction or damage. And so irregular connective tissue is best suited for that reticular layer. And you'll see that the reticular layer is going to be easily seen on any picture, even without all of this, as a little test-taking technique, because you can see that there's going to be this little bit of a plexus that we're going to learn about, or a little bit of a brady of blood vessels. And so you can always tell which part is which, typically on most of these diagrams, because the papillary layer is above this little plexus of blood vessels. And plexus in Latin means braid. And so this braid of vessels here. And so with that, we move on to our accessory structures. We'll get into a little more detail, but just to mention them: hairs, nails, exocrine glands. We saw epithelial tissues, and let's quickly revise that because that is part of our upcoming exam. So I like to review a little bit ahead of time of everything. And there's not a lot more to add. And so when we saw epithelial tissues, they make up epithelia, and then they make up glands. And probably one of the toughest concepts to teach you all in the beginning was to get you to grasp how in the heck do we go from epithelial to being something that secretes stuff, right? And so what you have to imagine is that you start off as something else, a different epithelial tissue, and then eventually there's going to be something we refer to as ducts. And so these ducts, when they embed themselves into the dermis, the epithelial tissue, the lining, begins to change into something entirely different. And in the case of integumentary system, what you're going to see is that you have on the surface that this is going to form stratified squamous on the top, the keratinized, and then it's going to dip into the dermis, and when it dips, you're going to have cuboidal cells, which are both epithelial cells, but you go from one epithelial cell into an entirely different one. Stratified squamous specializing in being a surface protective little guardian cell, and cuboidal serving as a secretion cell. So they change in functionality. So that's what we're seeing when we look at a few things. You're going to see that this is an example of here you have all this epithelial tissue, and then it becomes something else the second it goes inside of this gland, as we'll see a little bit later down the line. But it goes and it becomes something different. So even glands are considered epithelial tissue, and today we're only going to be seeing exocrine glands. Exocrine means that it doesn't need to rely on blood vessels. If something was endocrine, it would release the material out here into the dermis, and then all the material, the hormones, whatever it may be, will go into the blood vessels, into the capillaries, and then the capillaries will deliver it wherever it needs to go. But if it's just going to secrete it out through the duct, it's considered exocrine. You'll see another gland, and this is another perfect example of this. Here you have stratified, and then the second we dip here into this becomes a gland. This is going to be one of those hair glands or a sebaceous gland, but everything that's highlighted right now, it's a cuboidal cell in there. And so these cells will be exocrine because they secrete materials onto the hair, oil, onto the hair to keep it healthy and nourished. And so this is an example of exocrine. They did not need to secrete it into the capillaries, and then the capillaries didn't deliver it to the hair. That would make it endocrine, but that doesn't function that way. Exocrine means when things go from the gland straight out of the duct to serve whatever it needs to serve. Some of them are going to be obvious, like sweat glands, and one not so obvious, sebaceous glands, or hair glands. And so with that, we're also going to see nervous tissues. We're going to see some nervous tissues that specialize in different sensations, and that's what these black ones are going to be here. You can see that some of them are actually going to go into the epithelial cells. Some of them don't stop at the dermis. We're going to see others that never cross that basement membrane and stay just underneath it. You're going to see some down here that serve for different types of sensation, like pressure. These can detect things that are on the surface of the skin. The ones that are deeper detect things that require a little bit more pressure, but that's how you know you're being pushed really hard, because these are telling you you're being pushed really hard. And one that we all have experience with is this one here, the ones that make a sensation of pain when a hair is pulled out. And so that's what those are there. These are detecting pain in your hair when hair is being pulled on. And then we're going to have muscle. This is another form of muscles. Remember, we have cardiac, skeletal, and smooth. And this is going to be an example of smooth. Are we going to be able to control this one? No, we are not. And then we have another very popular form of our connective tissue, and a plexus. This word I like to introduce it early. And this is going to be cutaneous, meaning these membranes in the skin. And so this is a braid found within those cutaneous membranes of blood vessels. Make sure to put your phones on silence. And with that, next, when we go here onto the subcutaneous layer, it's going to be our hypodermis. As an FYI, this segment is not considered part of the integument system. It's actually not considered part of this system at all, but it is worth noting that it is underneath the dermis. And so sometimes it's naturally just called the hypodermis. And so it actually is in possession of two types of tissues that you also are aware of. And so it becomes areolar tissue. And one more that you should be able to recognize just off of pictures. What are those little yellow globs there? Adipose. So it has a combination of areolar and adipose tissue. So that's why during the exam, when I ask you about adipose tissue, I always say the best example you have is underneath the skin. Now the perfect answer is the hypodermis. But underneath the skin works just fine. And this is why you can hold on to some warmth because of your skin, because it has that layer underneath with adipose helping you retain a little bit of heat. And so do not forget, as a study approach, just learn this figure first. It'll be less overwhelming than it is to go slide by slide. Learn this figure first and then find a term or a way to describe each bolded term there. So some functions that are a little easy to understand and some that I'm going to clarify off of this table. Make sure that we know these functions. Some of them are going to be very obvious, like protection, right? If it's the most outer organ system we have, it's protecting us from abrasion, attack, infection with that areolar tissue that we have right underneath the epidermis. We have protection from fluid loss, right? If we have a little needle stab or anything, we're not going to just bleed lose water like a water balloon. And again, chemical attacks can be anything that's just on the surface. Your epidermis protects you by just shedding the lining of dead cells. We're able to secrete things from glands out that are coming through the epidermis onto the surface, such as water, like sweat, and that water usually contains salts. And then it also, in some areas, we're going to see that we have glands that secrete very stinky lipids, and that's why we have things like BO. So organic waste is like lipids, an example. Like lipids is an example. Like even when we think about, when I get to the fact, I don't want to waste too much time. Waste too much time. So maintain normal body temperature. This is going to be because of our fat, right? It allows us to insulate, and it also has capillaries or blood vessels that are helping regulate temperature underneath in the dermal region. It also provides us with skin pigmentation. And this is a very useful function for the body because it protects us from UV light. We have a love-hate relationship with UV light, as we're going to see in a little bit. And then it also produces keratin, and this is what makes skin such a durable material to protect us against abrasion, and also doesn't allow water to sort of just melt us. Water can somewhat seep through, but very minimal. That's why when you take long showers, you can see that your skin gets wrinkly, or when you're in the pool too long. But for the most part, you're not just going to melt away in water, and we have to be grateful for the keratin in that epidermis. And that love-hate relationship right here that we have with the sun and melanin, these 2 points you should tie together. because you need to be in the sun the same way plants need to be in the sun to get food. You need to be in the sun to get vitamin D, because it's going to be a vitamin that helps you metabolize calcium much more efficiently. And so vitamin D is essential in calcium metabolism. This long statement, just really, all you really need to know is that. Vitamin D, great for calcium metabolism. And what do you need for that? Sunlight or UV light, okay? But again, we have a love-hate relationship, as we're going to see in a second. And we store lipids in our fat cells in the dermis and underneath the dermis in the subcutaneous layer. And we're able to detect things that are affecting the integument system, such as pain. And in the context from an evolutionary perspective, right, pain is very useful for protection, right? You want to keep track of where you're getting hurt. You want to keep track of something is hurting you or going to cause physical damage to the skin. And then sometimes you need to be able to distinguish between things just by feel. And also temperature, right? To protect us from cold and hot temperatures and to always go towards very comfortable things. And everything that you're seeing, the last little statement here, relays that information to the nervous system. All of this, all of these nerves, anything that's classified as nervous tissue and anything classified as senses will always go from that surface to your brain, into your brain. You have two directions with the nervous system. You have a way going into the brain and you have one going out. Whenever we talk about senses, feelings, it's always going into the brain. Whenever we talk about movement, it's always going out to the muscles. But for now, all you need to know, these nervous tissues will relay the information into the nervous system, into the brain eventually. And so starting with the epidermis, it's going to be the most superficial layer. You can always define it by those little roller coasters that you can see on our little cake. And so there's a term to the base, the cell layer right above the basement membrane. The first layer that you encounter that is in contact with the dermis is called an epidermal ridge. And even if it's not highlighted for you there, it's always going to be the grooves that are always looping and in the layer of the epidermis, not the dermis, not the pink segment, but the layer above where the epidermis. So the segments that go into the dermis, these little segments, these loops, these roller coasters, these are our epidermal ridges. And to that same note, you're going to see another term that applies, dermal papillae. And so that's going to be the opposite. That's going to be the sort of thumbs up or the bottom part of that roller coaster here that's in the connective tissue layer where the capillaries are. And so you can see epidermal ridge, again, is going to be the border of the epidermis. And the dermal papillae are going to be those elevations or the boundary of the dermis, which is the layer in between. And remember, you have two layers in the dermis. You have what layer? Starts with an R. Reticular and papillary. Now, little quick sidebar, same concept, but sidebar. Now, when we look at our tissues, you may have noticed that in lab, we do not ask reticular tissue, but in lecture, you do have to know that it exists. And so that can be very confusing. Reticular tissue specializes in secreting what fiber? Reticular tissue loves to secrete which fiber? What is it? What are our three fibers? Collagen, elastic, and reticular. So it's called reticular tissue because it likes to secrete reticular. And our integument system, we're seeing this word again, reticular. But try to keep in mind that that has nothing, that is not related whatsoever. You have dense irregular, an entirely different type of tissue. They have no relationship between the reticular layer and the reticular tissue. Why you see the word reticular is that the word reticular is a descriptive word to mean a network, something that forms a net like a fishing net. And so stroma was the word to imply that very thing. And so the word reticular implies net-like. And so the reticular layer happens to be sort of be a net of material. This irregular tissue allows all of these little opening segments so that things like glands, hairs, blood vessels, more nerves, so that they can actually still intertwine through, but it's still, you still need that elasticity there. And so the word reticular again just means like a network. And so just be careful with that, because that's something that can be confusing. And so the dermis, my only point here was that in the dermis, you have two layers. You have the papillary layer and the reticular layer. You have your papillary layer and reticular layer. And maybe this can be confusing if I write them in the opposite order. Let's make sure I write them in the right one. All right. And so with that, our epidermis, the layer on top, which is keratinized, stratified, squamous epithelial tissues, lacks blood vessels. It is avascular. It relies on the dermis and all those capillaries in that dermis to provide oxygen and those nutrients that we've learned about, carbohydrates, proteins, lipids, nucleic acids. And they are the most metabolic demanding. And this has to make sense now if we think about this context here, that it takes 7 to 10 days for these cells to go from the lower layer, to go from this layer, the very base of the epidermis, to go all the way to serve as a little dead skin cell. It takes 7 to 10 days for them to really go from that bottom end to that top layer. And so that's what this statement means here. We'll see what this word means in a second. And so the cells in the surface layer, the ones on the most superficial, are going to be referred to as layer corneum. This word stratum means layer in Latin. As we see here, it's already here for you. Layers, strata. And so they are the most metabolic demanding because they are undergoing mitosis more often than a lot of the cells in our body. So they require a lot of nutritional input from the dermis. The primary cell now, you all learned it in lab as squamous cell. If it was called stratified squamous, each one of those little individual cells was called a squamous cell in lab, but now we have to add a little bit of a technicality. Each one of them is not considered a squamous cell now, it is called a keratinocyte. Site implying cell, and keratin because, the keratin because it secretes the protein keratin. And so our epidermal ridges are the boundaries of the epidermis. The dermal boundary is the dermal papillae, and this in turn creates what we refer to as an increase in surface area. And a lot of people don't never really understand what that means, but it's usually easier to use the fingerprints to describe that. And so our dermal papillae and our epidermal ridges create what you know as your fingerprints. So they create a unique type of surface with grooves that give us a little bit better gripping and stability on any surface. And that your epidermal ridges and dermal papillae result in your the grooves that you know as fingerprints. And every individual has unique fingerprints. Now, where things get slightly tough. Okay. Now keep in mind that we have not progressed into the dermis yet. We are still just in that superficial layer, the epidermis. There are 5 layers you want to log away. They are going to be stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, stratum basal. How many layers do we have here? 1, 2, 3, 4, 5. So on the previous slide, I know I glossed over. You don't have to flip pages. I'll show you. Here, thick skin and thin skin are going to be 2 different types of skin. This one only contains 4 of those layers that I just said. Thick skin contains all 5. So that's the note you need to have. When you have thin skin, you have 4 layers. You might have to flip pages, sorry. And in thick skin, 5 layers. And the last thing to add, the picture's cutting off here, soles. Soles, like the soles of your foot. And so you find thin skin everywhere in your body, absolutely everywhere. And the only place that you find thick skin is in the palm of your hands and the soles of your feet, the paws of a person. If you were walking on all fours, your paws, essentially, that's what I mean. That's what I mean for simplicity's sake. So thick skin is going to be, again, only in hands and soles. You see all 5 of the layers that we are about to talk about. Now, first one, stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, stratum basal. The one that you do not have in thin skin is lucidum, and I wrote that a little note here for you here. This is the one that's missing in thin skin, stratum lucidum. So there's always an order to these cells, and each layer there's always something different going on. And so if we start at the most basal end of this epithelial tissue, right? In case you've forgotten, this is the apical side of our epithelial tissue. This is the surface world, the surface of your skin. And down here is underneath your skin, right? You can see the epidermal ridges are here. And when we look at this segment, each one of this first layer here is going to be your stratum basal. That's going to be the first layer. It's even in the word, right? Basal, implying the first layer. Now, what makes this particular layer unique is that you have mitotically active stem cells. Remember that stem cells make more cells. So if you're a keratinocyte stem cell, you are responsible for undergoing mitosis, undergoing interphase, prophase, metaphase, anaphase, telophase, cytokinesis over and over and over again until your cell, the daughter cell that you created, is going to travel up and then eventually fill themselves up with keratin and die as a little dead cell. But you stayed over here to continuously do that forever and ever until your human being is alive. And that's the responsibility of the cells that are there. So they're typically considered basal cells, which just means a keratinocyte stem cell, which means a cell that's going to give rise mitotically to more keratinocytes. They are attached to the basement membrane. Keep in mind that hemidesmosomes, in case you need a picture, that when we went over epithelial tissues, we had here in this segment, desmosomes keep things anchored left to right between epithelial cells, and hemidesmosomes anchor things to the basement membrane, And in this context, you can see that this is our basement membrane, so our basal cells are clearly going to need hemidesmosomes to latch on to the deeper layer, which is on the basement membrane. And so that's the significance of these two statements. And lastly, we're going to see that we're going to have nervous tissues that go there, but don't worry about this. It comes up again in with a better picture, so don't worry about it here. Stratum spinosum. This is going to be named after the fact that the cells sort of look spiky or flat or thinner. And so the keratinocytes have now been created by the basal cells, and they've traveled upwards to the stratum spinosum. Now, the stratum spinosum, what's going on here, I sort of have this note that's a little hard to see, but what you want to write in here is that what you encounter there are newly formed living keratinocytes. You want to write also that you have melanocytes. That's an O, sorry, no, it's a little hard to read. Melanocytes. M-E-L-A-N-O. Comes up in the next page, I think, if I'm not mistaken. And then this last word, it says dendritic. And that little thing in parentheses says white blood cell. So let's rewrite that. Yes, white blood cell. And this one, dendritic, which I believe is already in your notes, dendritic. And so you have living keratinocytes that are migrated upwards. You're going to have a cell responsible for pigmentation. Pigmentation. And then dendritic cells, if it's white blood cells involved in immunity. So you can see even in your epidermis, we had mentioned that in the dermal area, in the papillary layer, it's made of areolar connective tissue, which has an insane amount of white blood cells swimming with all the fibrocytes, making all of this collagen and elastic. And so these white blood cells are defending you in the dermis, but you also have some in the epidermis, all the way up here in stratum spinosum. Ask away.
What's the word that starts with M?
Melanocytes, it's this word right. Oh, I thought it was on this page, but it's this one right here. Melanocytes like this. Melanocytes. Yes, and we'll see a picture of them right now, where they're at or anything, everywhere else. All right, so with that, so stratum spinosum is going to be responsible for containing dendritic cells, which are white blood cells. They used to be called Langerhans, but thank goodness we changed it to a more appropriate. Dendritic means that they have a lot of branches, and so these white blood cells are just making sure they're defending you from infection. Defending you from infection. And this layer, they're bound more tightly to the cells that are adjacent. So they no longer care about too much having hemidesmosomes, because hemidesmosomes anchor you to the basement membrane, but desmosomes anchor you to the lateral cell, to the cell next door. And so that's what really defines stratum spinosum, is that you have anchoring by desmosomes, more so than hemidesmosomes. Next, we have our stratum granulosum. That's going to be the next superficial layer. And that's going to be the boundary that you see here, that where we can barely see, or we can see the cell sort of, you can't really see nuclei anymore. And it's named after the fact that this is where the granules really start filling up with keratin and even start releasing out into the cell. And so the cells are no longer dividing, and now they are producing two proteins that characterize your skin. One is called keratin, and the other one is called keratohyalin. So the only one you've heard is keratin, but these are the two proteins that give skin their texture. And that's what granulosum does. Okay? They produce it. Now, lucidum. When you think about lucidum, this is a very high-powered image, and normally what you would see in a microscope, and I kind of wish it was consistent with what we would see in lab, but is that you'll see that this layer actually looks see-through. It looks see-through. And that's why it's called lucidum. It's like you would actually be able to see the back of the microscope slide, or through it. It would just be clear white, even though there's cells there. And that's why it's called lucidum. But what characterizes it is that this is where the cells begin to fill themselves up with keratin and die. They become our first layer of dead cells at stratum lucidum. And then finally we made it to stratum corneum. And so this is a segment where you're not going to really see any more nuclei. All the cells become flattened, dead keratinocytes that now serve as a little bit of dead cells that protect you on the surface of your skin. And so this has all 5 layers. So this is a picture of thick skin. And so you can see the difference between the two. This is thin skin everywhere else on your body. It doesn't have a huge layer of dead skin cells. But you can see that the thick skin on your palm and on the soles of your feet, you have a lot of dead skin cells, because these are the areas that encounter the most friction throughout your lifetime. But otherwise, you can see that even going up to here, even up till you get to that stratum corneum, we're missing lucidum. And so this up here is just corneum. And so all of these cells are still dividing, and there's not a huge layer of dead cells. So you can just see it just off of the pictures, the big difference between the two. And if you want an acronym for this, try to remember, come, let's get sunburned. Come, let's get sunburned. And you want to use the first letter here. Corneum, lucidum, granulosum, spinosum, and basal. I still use it all the time, and it's really useful. But if you're thin skin, what's the acronym? What does it change to? Yes, come get sunburned. Okay, there's a difference. So let's only exist in thick skin. Only in thick. And you remove let's or lucidum for thin skin. Now, before I come back to this page. So something that makes that layer stratum spinosum very unique, and even stratum basal, because you actually encounter both of these cells in those two layers. You encounter what we refer to as melanocytes, these large spider-like looking cells, and these are going to be located in the stratum spinosum. They're going to be located in the stratum basal. And so what you're seeing here is the dermis. You can see that this is going to be our basement membrane, and you can see that melanocytes exist sort of on that basal layer, and they have dendritic extensions into the spinosum. Now, I've mentioned previously that we have a love-hate relationship, and that's when melanocytes, or the narrative around melanocytes, comes to play. So within the basal layer, you have these melanocytes. You can see them. They're pigmented brown here. These cells are responsible for protecting your precious cells underneath in the dermis layer and all your organs, and protecting them from UV light. Like sunglasses, they sort of limit the amount of UV that's going to penetrate into the dermis by being a darker pigmentation. Because in case you're not sure how color works, because I noticed that, you guys might not taught that when you're young. When light, right, when you have white light, when you think about all light in general, you have to imagine what you see up there, white light. And when you look at white light, what you have to imagine is a rainbow in white light, because white light is every single color you know. Orange, green, purples, orange, green, yellow, it's all in white light. But when it goes, it travels, and then it goes to say, like this watch here that's blue, this leather strap is blue. And so why are you seeing blue? Because white light, like this pen, it's white, right? White goes and it hits this surface, this pigment, and what this pigment does is that it absorbs every single color except the one that you're seeing. So it absorbed green, it absorbed yellow, it absorbed red, it absorbed purple, but it didn't absorb blue. So the reason you see blue is because blue, being in that white light, goes and it was not able to go into the material, and then it gets reflected into your eyes, so you see blue. And so in this case, melanocytes, what they're doing is that they're protecting you from light in general, UV light. And that's because UV light can actually affect your DNA molecule. I'm just going to go ahead and just go to a picture just for the sake of telling a story, that you can actually alter these bases with UV radiation. Like if you want to actually do experiments on cancer, you just grab some cells, put some UV light on it, like the ones that you use to grow plants, and then you can make it develop into a cancer. And then you can actually make a cancerous cell in a lab. And that's because UV light goes to DNA molecules and it messes up the order of the bases, the instruction manual. And then so the cells lose the ability to undergo mitosis correctly. So if they lose the ability to undergo mitosis correctly, they can just keep going and they can't stop themselves from dividing. And if a cell can't stop dividing, that's why you get growths such as like a melanoma or skin cancer, little growths. So these little growths are like keratinocytes that can't stop dividing. So they're just making more skin cells, but they can't regulate themselves because UV light affected the cells, or the stem cells, the basal cells, from dividing correctly. So melanocytes protect you from that very thing. And so melanocytes protect you from cancer, but at the same time, your body needs vitamin D, and the only way you can get vitamin D is from being in the sun. And so melanocytes protect you from sunlight or the harmful rays of UV light. And that's what melanocytes are. And so the pigment that they produce is called melanin. That's the pigment. It's considered brown, yellow-brown, or black, and this gets placed into vesicles, and through the process of exocytosis, as you can see in our photo here, that these vesicles bud off of the melanocytes, releasing the pigment out into the keratinocytes, and that's why skin or keratinocytes can pick up that pigment, and then you have what we know as skin color. Some populations of human beings have more darker pigmentation because their branches are a little more extensive and the secretion of melanosomes is a little bit higher. And this is typically because if a population of human beings was from, say, more arid and desert areas like Africa, a darker skin pigmentation is more advantageous in an area where there's not a lot of shade. And if there's not a lot of UV radiation or a lot of more foresty, higher altitudes, maybe they only get sunlight a few hours a day, tended to not need as much melanocytes protecting them because they didn't get as much UV exposure. And they still needed a good healthy amount of vitamin D, so melanocytes would be very inappropriate because vitamin D is so precious when you don't get enough sunlight. Ask away.
Does thick skin have a lot of melanocytes?
No, right? Because when you look at some individuals that have darker skin color, if you look at their palms and at their hands, you can't see the melanocytes because those melanocytes are so deep, and that stratum corneum, the layer on top, there was so many dead cells that you can't even see all the way down to those two layers where you would encounter this, which is, again, in the stratum basal or the stratum spinosum layer. Here we go, stratum basal layer, which is the most deeper layer. And that's really it. And also, as a little fun fact, I'm not going to test you on this, but everybody has the same amount of melanocytes. No matter what race you are, the only difference is that the branches are different between races. That's the only difference. That's the only thing. But you have the same amount of these melanocytes for the most part. Every person does. And you don't need to worry about this pigment and where it comes from. You just need to know that keratin is another pigment, just like melanin is. Melanin and keratin are two skin coloration pigments.
So, Professor, so people with albinism, or people who are albino would not have any...
Yeah, there's an issue with the melanocytes, a genetic predisposition to not have wild type. That means normal. You can't really say that, but not to have what's commonplace. It's typically something wrong with the, not wrong, something different about the melanocytes. And since we're still on skin pigmentation, the effects of blood supply, the blood supply has effects on skin color, and we learned about this little plexus that separates our papillary layer and our reticular layer that lies underneath. That is called our subpapillary plexus. And so more blood flow is what makes someone redder. You can think about blushing, makes it a little bit easier to understand. And a lot of times when it's really cold and someone's really pale, it's typically because, and most of the time when it's cold like today, right, if you're in a room if you're somewhere that's very cold, your blood will leave your extremities where you don't need as much blood supply as opposed to where you do need a blood supply, which is your organ system. So when it's very cold out, your blood likes to pool into your center trunk, and then that's why your hands get all cold, and that's why they get all pale. And it's because blood supply leaves the limbs and goes to serve the trunk of the body instead. And so turning bluish because of a lack of blood flow is called cyanosis. So you can see how blood supply can affect you in a few ways. You can be red, you can be pale, or you can be blue. And later down the line, we're going to see that sometimes the color of your skin can indicate even what's some contents inside of your blood. Like if you have a very active liver or something wrong with your liver, you can turn yellow. And we'll learn that a little bit later down the line. And that's really it for this page. All right, good news is that there's not much more to add to this dermal layer than what hasn't already been said. The epidermis does come with a little bit of complication, so try to remember that the dermis has two layers, papillary and reticular. The epidermis has either 5 layers or 4 layers. Depends if you're thick or thin skin. Now, the dermal layer The papillary layer is named after those little thumbs up, the little boundary of the dermis, the one that goes upwards, the dermal papillae, these little grooves that go upwards. And so that's going to be our papillary layer. It is made of areolar tissue. We know that now. We see that it contains capillaries, lymphatic vessels. You do not need to worry about it. I am just going to keep it in the cardiovascular segment. But it does contain sensory neurons. Neurons responsible for sensation. And that's what these structures in black are. Reticular layer, again named after the fact that it's interwoven meshwork like a net-like structure. It does not have reticular tissue. It's an easy mistake to make, but it instead has dense irregular connective tissue, which is heavy in collagen and a little bit of elastic, and that's why it has a bit of a rubber band feeling to it. It also contains blood vessels and lymph vessels, and it contains nerve fibers just like the layer on top. And this is where we see a lot of other accessory organs originate, such as follicles or the hair, and also glands. And underneath we have our hypodermis, which contains areolar tissue and adipose tissue. Adipose tissue. Now, our nerve endings, we have different types of feelings and sensations. So learning these 5 are important. You'll notice we have free nerve endings, and then we have some word that are going to have the term in common called corpuscles. When it has the word corpuscles, to you as students, this word means capsules. Capsules. And so you can tell that some of these have little capsules on them. And that's all a corpuscle means, is that it has little capsules. And so free nerve endings do not have little capsules, and these are actually embedded within the epidermis. You'll notice that the rest of them are either bordering or only in the dermis, but free nerve endings are letting you know when you're receiving damage to the skin. That's why little cuts hurt, paper cuts, things like that. That's why they hurt. You have nerve endings detecting sensation on the skin. Not only are they able to detect pain, but they can detect finer touches and even some light pressure. And then you have another one called tactile corpuscles. These are going to lie a little bit underneath, and these are not going to be in the epidermis. You'll notice that they actually exist only within the dermis, and that's what makes them unique from free nerve endings, and they detect texture. And you're not sure what this means? If I gave you sandpaper and then a silk blanket, you're able to tell which one's which by just feeling. That's what being able to detect texture is. That's even what tactability is. Tactability, if I gave you some quarters, some pennies, some nickels, and I put them in your pocket, and if I tell you to close your eyes and give me the quarter or the penny, you can probably figure it out just by the size, by the texture, and that's what these do, the tactile corpuscles. Fine detail. These sensory, and I think, are my notes correct? Because this feels a little off. These notes are off, actually. Shouldn't say corpuscles, this should say discs. Even as I was talking right now, something didn't make sense to me. Because these are all found in the epidermis, and this is how I organize it in my head, is that these usually go into the epidermis. So these went into the epidermis, and the only other ones that do go to the epidermis are these tactile discs. So I'm just realizing this should have been discs. But their function is the same thing, which is good news for us. Now, Meissner corpuscles, another word for this one is going to be tactile corpuscles. So be careful with that. Discs are found in the epidermis. They are found in the epidermis, while these corpuscles are only found in the dermis. So yes, tactile discs should have been there. And these here should be tactile. You'll sometimes hear Meissner's because it was named after the person who discovered it, but then now they're changing it to more practical words, and this one's going to be lamellar, and that's it. And so these Meissner corpuscles are going to be in the dermal layer detecting fine touch, light touch, vibrations and pressure. The Pacinian ones, let the P remind you of pressure, that they detect deep pressure and vibrations, because look at the locations. These are on the surface of the skin, so they have more access to more finer detail. These need to be pushed much more further for them to even feel anything. So that's why we say they detect pressure. Ruffini, you can tell that your skin is being stretched because there are receptors that are sort of keeping track of the regular, or they're embedded within the collagen fibers, and they're detecting how much they're being stretched. And so these are called Ruffinis. The new word for them is bulbous corpuscles. You can use, I'm going to put both on the exam, so learn either one.